This is Dr. Gilchrist with a screencast to finish talking about these different neuroimaging methods so that you have a better understanding of what we're talking about here. So let's do a quick comparison of MEG, which we talked about last time, which measures the magnetic fields that are produced when neurons fire, versus EEG, when we actually get the electrical activity produced by those postsynaptic potentials. So the temporal resolution of these is actually the same. They're looking at the exact same thing. Now, because of the orientation of neurons, the way that they're kind of aligned, as well as the right hand rule, MEG is often better if you're looking in deeper areas, such as a sulcus. So it's better for sulci activity. EEG are usually better for the raised portions of cortex. Oftentimes, we will actually find that MEG is really common in neurolinguistics, and that's because the inferior frontal sulcus and the superior temporal sulcus, Broca's area and Wernicke's area, are both really important for language. So now moving on, let's talk about a different method known as transcranial magnetic stimulation. And so generally, we might want to have an instance where we actually stimulate activity. So what we do is we take a small wire coil and we actually pass pulses of electricity through that coil, thus creating perpendicular magnetic fields. And the idea is that these magnetic fields can be used to stimulate different portions of cortex, in addition to potentially interfering with functioning and temporarily impairing that function. Now, usually we will use TMS to impair that activity, but sometimes we do want to facilitate it and enhance it. Typically, what we will find is that these magnetic fields will either cause many neurons to fire, or if it's repetitive, it can actually inhibit a neuron. And typically, what we will find is that you will have very characteristic muscle twitches or speech arrest, depending on where this is applied. So what's kind of cool about TMS is that it does produce temporary lesions, or it can facilitate activity. So if you want to determine if an area is really important for a particular cognitive process or task, you can do something like this. We do know that repetitive transcranial magnetic stimulation, typically you are repeatedly inducing this stimulation that produces longer lasting changes in the brain. It's still pretty experimental, but it has been identified as a potential treatment for things like depression, obsessive compulsive disorder, and so on. So now we're going to talk about direct techniques. We talked about EEG, MEG, and we've talked about single cell recording and TMS. So now we're going to talk about ablation. Ablation is by and large the oldest method in neuroscience that has been used to actually look at the functions of the brain. Now, this is something that we do in both humans and animals. Obviously, with humans, we don't intentionally ablate brain tissue, but we do actually study patients with existing lesions to understand the functions of those damaged brain areas. Now, just in case you don't know, a lesion is an area of damaged tissue, and ablation studies may sometimes be referred to as lesion studies. Now, one major problem with lesion studies is that any one brain area doesn't really act in isolation. It's connected and communicates with a lot of other brain regions and networks. So just because a brain area is damaged and a particular behavior is also impaired doesn't necessarily mean that that area is responsible for that behavior. It could be another connected brain region that supports that behavior, or it could be multiple brain areas together. So there are two major ways that we can produce lesions. You generally want to be precise when you are dealing with animals. All of these are accomplished by anesthetizing the animal putting it into a stereotaxic device, which I showed you last time, that helps stabilize the head and it allows you to be precise. So the first one we're going to talk about are radiofrequency lesions. These are basically produced by inserting a wire through the brain in the area you actually want to ablate. You inject a current, and the heat from that current actually damages the tissues, including neurons and glial cells. Now, this is effective, but it's not very specific. It's actually quite crude. Generally, you will find that this will produce more area damage than you actually want. Here's a quick example of what that looks like. This is in the brain of a rat. You can actually see that the arrows are pointing to the area where that lesion actually took place. The other thing that we can actually do is something called an excitotoxic lesion. This allows you to be more precise. So what you do is you inject through a very narrow tube that we call a cannula. We usually will inject an excitatory amino acid neurotransmitter, something kind of like glutamate, but not actually glutamate, but it will bind the glutamate receptors. And because of that, that will produce EPSPs, but at a rate that actually ends up destroying the cells. Too much amplification will kill cells as it destroys the balance of the ions, it damages the ion channels and receptors, and that will cause the cells to die. Generally, we prefer this. It's a little bit more precise because it doesn't destroy all the passing axons from other brain regions, but just neurons in the injected region. And then finally, another method that allows us to target specific types of neurons is by targeting ones that produce particular NTs, neurotransmitters. So 6-HD is actually a chemical that's very similar in structure to dopamine as well as norepinephrine. The reuptake transporters for dopamine and norepinephrine can't tell the difference between 6-HD and the neurotransmitters, so it binds the receptors and the reuptake transporters will take that up. Once inside the cells, it produces free radicals, which end up damaging the neurons. Now, this is just one example, but there are other drugs out there that will affect other types of neurons in a pretty similar manner. Now, for any one of these ablation surgeries, it's really important to take into account damage that you actually do by anesthetizing an animal, drilling into its skull, inserting a cannula. Any of these could produce damage even if I wasn't injecting a radiofrequency current or a type of drug. So if we want to assess the relationship between a brain region and behavior, you have to control for those effects. So typically what is done is called a sham lesion or a sham surgery. So some animals will go through all of the same surgical procedures. They get anesthetized, they cut into the skull, they insert the cannula, but they don't get the toxin or the radio frequency current. And then you compare the sham and the actual lesion group, and if there are behavioral differences between the two, you can at least conclude that it's not the surgical procedure producing those differences, actually the brain damage. So generally, in order for ablation surgeries to be accurately performed, the head has to be put into a coordinate system that has all of the different regions of the brain mapped out. So we can locate specific brain regions for each animal by putting them in a stereotaxic apparatus, which allows the experimenter to locate numerical coordinates in X, Y, and Z planes on the animal's head and drop a cannula in that location. During surgery, we're going to locate a point on the skull that is called the bregma. That is the junction between some of the bones of the skull. And when the bones fuse, they form sutures that actually look like seams. The junction between the sagittal and coronal skull sutures is where the bregma is, which you can actually see demonstrated right here. So here's our sagittal suture, here's our coronal, and the bregma is right here. Now, here's what this would look like. This isn't just used for animals in surgery, but it's also used in human surgeries. Neurosurgeons might use a combination of coordinates and MRI scans before removing a tumor or damaged tissue. We can also use these stereotaxic coordinates to place a stimulating device or an implant to record electrical activity. This is often done in animals when we want to record neural activity once they're awake and behaving, or this can be done in patients with epilepsy. Now, generally, after an ablation surgery has been performed, it's important to verify the location of the lesion when you report the results. You want to make sure that you aren't off the mark, given that every brain is different, every skull is different, and they're going to be a little off the stereotaxic map. So typically to do this, scientists will fix the brain. They'll replace the blood with chemicals like formaldehyde that preserve the brain tissue and actually allow the brain to be handled and not just fall apart when you slice through it. So it hardens it and prevents it from decomposing. You then take slices of the brain according to those stereotaxic coordinates. So you have all of these little thin slices with labels for where they're located in the brain. And at that point, you stain them. Now, sometimes you might want to stain cell bodies. And then finally, you view these under a microscope to determine the exact location of the lesion and the size of it. And generally, what you will do is are the results, not literally, the results of animals whose lesions were too far off from the rest of them. So now we're kind of moving on into our neuroimaging methods, fMRI and PET, as well as CAT scans and MRI. So generally, we're going to move on to neuroimaging. I've said generally a lot today. There are two major branches of neuroimaging. There is what is called structural neuroimaging, where we're actually looking at the structure of the brain. So this would be for things like identifying tumors, aneurysms, or strokes. And we can either do that through a CAT scan or magnetic resonance imaging or MRI. For functional neuroimaging, we're actually getting an image of the activity. So we look at things like metabolism, blood flow, or receptor binding. And the two examples that we use for that are fMRI and positron emission tomography or a PET scan. We're going to start with a little bit of computed tomography or CT scans. Sometimes they're called CAT scans. That's what I call them. These have been around since about the 1970s. So during a CAT scan, you lie on a table inside of a donut shaped machine. An X ray tube inside of the machine will actually rotate around your body and send small doses of radiation through it at various angles. So the X-rays are going to pass through your body, and different types of tissue will absorb different amounts of radiation. You have detectors inside of the scanner that measure the radiation that passes through your body and converts it into an image that shows the different tissues of the body, including the brain. So CAT scans help build an anatomical picture of part of the body under investigation. So here's a quick example of what CAT scans look like. Here's an example of the kind of image it produces and how it kind of works. So in this case, that particular X-ray is going to circle around the body, and it's going to basically be, these X-rays are going to be absorbed at different intensities depending on the tissue. And we'll talk about that more pretty soon. In fact, right now. So what we're looking at here are Hounsfield units. This measures how much of the X-rays actually pass through or how little is absorbed. So in general, air is not dense at all. You get absolutely no absorption with X-rays. On the other hand, fat is a little bit more, water is a little bit more, brain tissue, both gray matter and white matter. And at this point, we get to things like bones, which are more solid, and as such, they're going to absorb much more radiation.